Hello, and welcome to another exciting episode of Let's Watch a Video Instead of Doing the Lab in Person. In this week's video, we are going to be looking at the histology for the special senses. Our first slide is the retina, where we're interested in the photoreceptors, which are called rods and cones. Now, this view, this is anterior, and it's posterior back here. So I'm going to draw an eye. So here's the eye. There's the cornea. Here's our iris, our lens, our ciliary body. And we have our retina back here. And we have the sclera, that's the outside part of the eye. What we did is basically took a little picture of this retina, and we can see it over here. Now in this view, we can see the sclera. It is dense, regular connective tissue, and that's actually part of the dura mater. We have our choroid coat right here. Then we have three layers. So we're basically looking from sclera, choroid coat is in there, then the actual retina, which is what we're interested in. The first layer, the deepest layer, the layer that's right up against the choroid coat, is our rod and cone layer. And there's lots and lots of rods and cones. I could sort of see there's a cone-shaped thing there. I could sort of see there's a rod-shaped thing there. So this is our rod and cone layer, right in this part of the eye. Now, rods and cones are connected to bipolar cells. So this would be the bipolar cell layer. And I can tell this is the periphery because there's not that many bipolar cells compared to these cells. And you have mostly rods, and the rods converge on bipolar cells. And then our third layer, we have ganglia cells. We don't have very many ganglia cells here because the bipolar cells are converging on them. So let's draw a retina. So we're going to be going back and forth between this picture in your lab book and my drawing, and actually, it's going to actually end up looking something like this. So I'm going to start with drawing the sclera, which is dense regular connective tissue. And I'm going to draw it sort of as a circle. And let's do it a little bit thick. And here's our sclera. Yes, it magically changed from nice and colorful to orange, which is more like the real color. Next, we have the choroid coat. That's the vascular layer. So I am going to put my choroid right there. The important part is the retina is made up of three layers of cells. We have the rod and cone layer, we have the bipolar cell layer, and we have the ganglion cell layer. So we're going to start with the rod and cone layer. I'm going to draw some rods in here. Here are the rods. Now on the actual picture, you can't really see the rods that clearly, but you see all the nucleuses. And they're so packed in, you can actually see the nucleuses of the cells behind it, another layer deep. So you're going to have this big, thick layer of nucleuses here. Now I want to put in some cones too. There's three kinds of cones. There's red cones, there's blue cones, and there are green cones. And what you see on the histology slide are all the nucleuses. And see how I made the cones stacked on top of each other? That's actually how the rods are. So you just have this really, really thick layer of nucleuses right here. So this is the rod and cone cell layer, and this is their nucleus layer. We have our sclera, we have our choroid coat, and then we have our rod and cone cell layer. You have the photoreceptors here sticking forward, facing the choroid, and then you have the nuclei. So label these structures on your picture. Our next layer is our bipolar cell layer. Now, bipolar cells look like this. They're going to have a central cell body, an axon, a dendrite, and an axon. Now, they are going to be different. Over here, where the rods and cones are, our next layers are Next layer of cells is our bipolar layer. And remember, the rods and cones are very dense. They're very dense as well, but it depends on where they are. If it's over where we only have rods, like the periphery, you're going to have fewer bipolar cells than you would here because rods converge on bipolar cells, cones do not. So remember our bipolar cell, we have our cell body, we have our dendrite. And this dendrite could be attached to a few cells. And this one's going to be attached to a few cells. Sorry for misspeaking there. So then we have the axon going the other way. And again, since these are the cell bodies, you're going to have nuclei in all of them. So you'll see a lot of nuclei on the histology slide. Now the bipolar cells for the cone layer are going to be less dense, but look at all those cones. There's a lot more cones in there. So again, it's going to be this big mess in here where you have lots of cells on top of each other. We have our retina here. We have a very dense layer of rods and cones. I should probably add a few more layers of nuclei here. Then we have our bipolar cell layer. So you still have a lot of nucleus all on top of each other, but it should be a little bit less dense than this. And I could always tell this is the rod and cone layer because I have my sclera here, and I have my choroid coat there. So label the cell bodies of the bipolar cells and the bipolar cell layer. This is what it looks like on the real histology slide. So here we have the sclera. Here we have the choroid. Then you have rods and cones, and there's a couple cones here. They do look conical. Then we could see these rod-shaped rods. Now this has been blown up 1,200 times at least. I could see my very dense nuclei of the rods and cones. So the rod and cone nuclei layer is extremely dense here. Now over here, I have my bipolar cells. Because this is mostly rods here, I have a thinner bipolar cell layer. Again, they're all on top of each other still. So there's probably half as many cells here. Then our third layer, since it's converging, and this is only rods, is our ganglia cell layer. So let's draw that next. So the ganglia cell layer for the cones is actually going to be really dense because you have one cell to one cell to one cell. Over on my periphery where I have rods only, it's going to be less dense, so I only have a couple. And they're actually connected to several axons. So I'll only draw a couple, and it's connected to a few. So it's very sparsed out. Over here, I'm going to have a lot more just because it's a one-to-one issue. But still, there's going to be less than you would have otherwise. So these are the ganglia cell layers. And again, they need nuclei. So that's the ganglia cell layer. So make sure you have all of this labeled on your histology. And we will then go on to the next histology. And again, for the lab exam, I will give you a picture that looks something like this, and you'll have to identify. So on the histology slide for the exam, I'll give you a picture like this, and you have to identify the choroid, the rods and cone cell layer, the bipolar cell layer, and the ganglia cells. And this is pictures actually what it also looks like, so you're welcome to try to draw either of these pictures as well. I'm going to skip to the taste bud histology now. And taste buds are found on these structures called papilla. Papilla are just the bumps on your tongue. And there's different flavors. There's folate papilla that have their taste buds on the side here. There's my favorite, the valate, V is for victory, that also has its taste buds on the side. The fungiform, that's towards the front of your tongue. Those have the taste buds on the top surface. And then we have our 80s punk rockers, the filiform, that don't have taste buds at all. So we're going to draw a single taste bud. So the taste buds are in here. This would be a folate taste bud, or folate papilla. And these are my taste buds. And there's actually little tiny taste cells in there. So let's draw this. So we're going to be drawing this taste bud right in here because he looks cute. And basically we have the crypt right there, formed by the papilla. And there's a little tiny pore. So we'll label it pore. And the gustatory cells in here are going to have little tiny hairs sticking out of them. So let's try the little tiny hairs sticking out. And these are actually attached to all the cells. So we have our pore, and we're going to have gustatory cells. Now the gustatory cells are these elongated cells that are basically connected to the pore, and the hairs come out. And I'm going to draw them in different colors. And they'll end up looking something like that. So they're all clumped together. Now at the bottom are some basilar cells. And they think these cells are going to become gustatory cells eventually. But they're not ready yet. So basically the gustatory cells die, and they're going to be replaced by these basal cells. And that is your taste bud. We're now going to draw the olfactory epithelial real quickly. Actually, I just misspoke. We'll do the olfactory epithelial and we'll do hearing in our other video. So thank you for tuning into part 1 of the current episode of Let's Watch Videos Instead of Lab. Please watch the second part. My sponsors need